Thank you. Thanks so much for the opportunity to talk. I'm just going to talk about our work using nanoparticles for rapid diagnostics. And so we are all highly familiar with infectious diseases because we're all personally impacted by COVID-19. But I do want to remind everybody that infectious disease is a global problem even before COVID-19 hit us. So this is a map from the National Institutes of Health, from the NIAID, showing the number of infectious diseases that are currently a problem in the world. And remember, this is pre-COVID-19. So there's many diseases that are newly emerging, like Hanta, things that have been re-emerging, like Zika, and things that are even engineered. And so this is a map to show you the scale and the number of infectious diseases that are a problem. So we know that diagnostics are badly needed for COVID-19, and this is true for every infectious disease. And this is due to the fact that many diseases have initial symptoms which are very nonspecific. So things like fever or nausea or headaches, okay, these are common to lots of things, even the common cold. So diagnostics are needed to provide, basically a diagnosis for a patient, and these aid in helping confine the epidemic, as well as quarantining, so whether or not a patient needs to be quarantined, as well as to guide treatment, and then also for disease surveillance to basically map where the disease is spreading. Okay, outside of coronavirus, this is really important for co-circulating diseases. So Zika, Dengue, and Chikungunya are infectious diseases that are spread in tropical areas. That's due to the fact that the vector for these is this mosquito, the Aedes mosquito. These are very important. For example, dengue has a disease burden that's in the billions of people, okay, with over hundreds of millions of cases every year. So they all spread in the same areas because these mosquitoes are in the same areas, and they all have very similar initial symptoms. So Zika and Dengue and Chikungunya all have initial symptoms of fever or sometimes a rash, and so forth. Okay, however, the outcomes of each of these diseases are very different. So Zika, we know, is tied to microcephaly and also Guillain-Barré. Dengue can result in the hemorrhagic fever, which is basically the hemorrhaging of your insides. And then finally, chikungunya can result in arthritic pain. So you can see that if you're confronted with a patient who has these initial symptoms, how you're going to treat them is really different depending on what they've been infected with. So you need a diagnostic to be able to distinguish between these infections. Now, currently, the state of the art for diagnosing infections is PCR and ELISA. These are methods which are highly sensitive and specific, and they've been used successfully all over the world. However, they do have limitations in that they have a very slow turnaround for readout. So even in developed countries for COVID-19, like here in the States, it's sometimes you have to wait several days for a COVID-19 diagnostic result. And this is due to the fact that there's just a limitation in how many of these you can run. So these instruments need to be operated by trained personnel. They require special reagents. They have to be done in a centralized lab, so a fixed location. And there's also logistics in terms of just supply chain management of their cold chain and the reagents. Okay, and these will either detect the virus itself, or in ELISA, it actually can detect antigens or the antibodies that are produced in response to them. Now, the World Health Organization issued guidelines for how diagnostics should be, and this is kind of like an easy to remember acronym called ASSURED, and they said that diagnostics need to be affordable, sensitive, specific, user-friendly, rapid, equipment-free, and deliverable. Okay, and this is kind of like the holy grail for all diagnostics, and it's important to at least try to hit most of these. You can see already that PCR and ELISA, it's difficult for them to hit many of these because they're not cheap, they're hard to be equipment-free, and so forth. And so an alternative format to detecting for disease diagnostics is the lateral flow assay. And lateral flow assays we're all familiar with because this is the same format that's used in pregnancy tests. So you add, it's very simple, you add a fluid that goes through it, 2 lines is positive and 1 line is negative. And just to remind everybody how these work, if you have a biomarker in the sample that you're looking for, you add it to this paper strip, and it wicks through due to capillary action. So no power is required. Then if that biomarker is present, it will bind to nanoparticles with an antibody or some species that can bind specifically to that biomarker. This complex wicks through, and then deposited on the test area is another species that can also simultaneously bind to that biomarker. So what it does is it accumulates gold nanoparticles at this test line. Okay, and then there's a control area that has antibodies that can bind to the nanoparticle conjugate itself, and this gives rise to another red line here. So nanoparticles have a very strong optical absorption, okay, gold nanoparticles in particular. So this is what gives rise to the red color that's present you see in this strip. Now you can see right away that these tests have the potential to meet many of those requirements from the WHO. Okay, we know that these are rapid, so they go from sample to answer in 15 minutes, so you can get an answer right away. They're very robust. You can stick these in your pocket and carry them around. You can do this anywhere. It doesn't require a lab, and all the reagents are self-contained in the test. Furthermore, these are made of paper. Paper is easy to make at scale, and so it's very cheap to manufacture, and it's very easy to use and can be operated by non-experts. Okay, so you can see right away that these have the potential to make a big difference as a diagnostic tool. Okay, now what happens in the test that results in this colored test line is basically an antigen must be bind to, that you're looking for must bind to simultaneously an antibody that's immobilized on the test area, as well as on the nanoparticle itself. Okay, and it's this double binding event that gives rise to that signal. Okay, I do want to point out that this is the cartoon that everyone always draws for these tests, this double binding event, but in reality, it's not as simple. Okay, we know that when you put proteins and DNA on surfaces of particles, those proteins can denature, they can be oriented incorrectly, they can be sterically hindered by the surface chemistry of the nanoparticle, so all of these can conspire against antigen binding. Then there's surface effects due to the nitrocellulose. So when you immobilize that antibody on the surface, it can also be obscured by the surface as well. And furthermore, nitrocellulose is porous, so it can actually trap some of those antibodies or antigens in it. Okay, this whole entire thing is under fluid flow, not like at rest in equilibrium in solution. So this is a transient effect. It's not really an equilibrium binding event. And then moreover, this fluid is a biological fluid of some sort, urine or blood or saliva. And there's proteins that are present at very high concentrations. Okay, and so this can result in a protein corona that will could potentially prevent this binding event to occur. So you can see there's a lot of the surface effects that are at play here. So in my group, what we do is we study this sandwich immunoassay format for nanoparticle antibody conjugates. We study many aspects of this. So we do things where we vary the nanoparticle size, material, and shape with the goal of either introducing enhancements or increasing the sensitivity of these tests. We study the protein corona that forms around them in these lateral flow assays. We vary things like nanoparticle surface chemistry and conjugation, and we engineer different devices. So today I'm going to just tell you one story about how we take advantage of the well-known property of nanoparticles, basically the ability for them to be made in different colors, to impart new capabilities to tests that are not possible before. Okay, so I do want to start with how we decide to make these tests. How do we decide, what antibodies and what biomarker to go after? So for every disease, we have to pick a biomarker. And if we are thinking about flaviviruses like dengue, okay, there is this biomarker, which is called non-structural protein 1, which is very convenient. And it's known that this biomarker is secreted at very high concentrations early on in infection. So if you're infected with a virus, very soon after that, your body starts to produce this protein NS1. And so this is also produced at very high concentrations, which other people have already characterized. So this makes it a really good candidate as a biomarker for infection. Note that it also kicks in before your immune response does. So it's a good way to determine early on if you've been infected with dengue or other flaviviruses. Okay, now traditionally what happens is that if you have a biomarker of interest that you want to make a test for, you have to be able to get antibodies for it to make that immunoassay. And so if you were to do this on your own in a lab, this is the procedure, and it's actually quite labor intensive. So you challenge animals with that biomarker, you generate antibodies toward it, you grow clones in hybridomas, and then of hybridomas, and then you test them for binding to the antigen using flow cytometry. So this is a labor-intensive process just to narrow down the antibodies. But remember, when you run them in a paper assay, they have to bind in a sandwich format. So you have to have a simultaneous binding format. So then you need to take all of those antibodies and screen them in what we call pairs. So put all of the antibodies on the paper, put all the antibodies in the nanoparticle, and then run them with the antigens and see which ones bind to the ones to the biomarker of interest. Okay, so this is a labor-intensive process. Okay, traditionally, antibody production for a new antigen would take anywhere from 16 to 24 months and cost hundreds of millions of dollars. Okay, recently with COVID, we've seen a great acceleration of this process, but it's still very long. Okay, and the problem is that because of this, it takes almost a year sometimes to produce a rapid antigen test. So if we look back to Zika and Ebola, it took almost a year to produce these rapid tests. If we look back to the onset of the disease when it first started, cases were multiplying in weeks, right? So this time scale to make a new test is too long, okay? And so this is a major bottleneck in, in making these tests, is antibody production. Okay. Now, however, there is an opportunity if you think about cross-reactivity. So we know that sometimes antibodies are cross-reactive. In other words, they bind to things that are off target. And typically this is viewed as a bad thing. So for example, if you take a commercial dengue test, some of them will actually be cross-reactive for other flaviviruses. So if you run this dengue test, you get a positive test. So two lines is a positive. But if you actually run it with Zika, it also can result in a positive test, as well as yellow fever and also West Nile. Okay, so normally this is a bad thing because it means it's cross-reactive. But if you think about it, it is also an opportunity. So for example, pretend it's 2015. Okay, you don't have a Zika test yet. This dengue test actually will still pick it up. Okay, so there, you can somehow use this dengue test to be able to pick up yellow fever or to diagnose it. However, you still have to be able to differentiate it from the original test because you don't want to know, you want to know if you've been infected with Zika and not dengue. Okay, so how do you do this? And so what we're going to show is a strategy to use nanoparticles of different colors, okay, that will allow you to repurpose antibodies that were raised for a different antigen for a new one. Okay, and this has a potential to be used as a point of care. If you have this strategy, then you can use this as a way to generate a rapid antigen test very quickly. Okay, before the time that antibodies are produced. Okay, so this means you can use a stockpile of antibodies that you have. Okay, so what we decided to do was to ask this question: could we take dengue and Zika antibodies? Okay, these are antibodies that were raised specifically for dengue and Zika, and repurpose them to pick up yellow fever. Okay, so we're going to pretend that we don't have yellow fever antibodies, so we want to use these other ones to do this. Okay, if we look at the gene, the phylogeny of flaviviruses, we can see that dengue and Zika and yellow fever are all somewhat closely related. So there's a potential for cross-reactivity. If we compare the sequences, okay, for NS1, for dengue, dengue 2, and Zika, we can see that there's high overlap with yellow fever, okay, on the order of 55%. Okay, so this also confirms that there's high potential for cross-reactivity. So then what we do is we try to find cross-reactive antibodies, okay, raised for dengue and Zika that will bind to yellow fever. Okay, so we have a small pool, just 3 off-the-shelf antibodies, 1 monoclonal raised for dengue, a polyclonal raised for dengue, and a polyclonal raised for Zika. And we use polyclonals on purpose because they have the potential for higher cross-reactivity. And we test that to see if they are indeed cross-reactive. And we do ELISA, so we basically use these antibodies to bind to dengue in green, Zika in blue, yellow fever in yellow. And we can see that the dengue antibodies bind well to dengue, but they also do bind to yellow fever as well. And same for the Zika antibody. Okay, so these antibodies are cross-reactive for yellow fever. Then what we do is we screen these in pairs to see if they can bind to yellow fever. So again, what we do is we take a subset of our antibodies, just the polyclonal ones, we paint them down on the test line, okay, at this location. We take those antibodies as well and conjugate them to our red nanoparticles made of gold, okay, and we run them in pairs and we see which ones bind. Okay, and we can see that sometimes we get binding, sometimes we don't. And we tabulate this all in this basically table with a heat map. Okay, so higher intensity and stronger color means that it bounds more strongly. Lighter intensity or lower numbers indicate they don't bind as well. Okay, and we can see, more importantly, that some combinations will bind to yellow fever. Okay, some combinations will not. Okay, and so there's a pattern differential already. Then what we need to do is to make a nanoparticle of a different color. And so what we do is we take advantage of gold nanostars. Again, gold nanostars are very easy to synthesize. It's basically gold chloride with a Heaps buffer, and this results in gold nanostars. And if you tune the Heaps concentration, you can tune the shape of the nanostar that results, shown here, by growing longer and longer arms. Okay, and this results in a shift of the surface plasma resonance, shown here, okay, to the red. So you can make lots of different colors in this way. And then what we do is we go and choose the correct, the colors that have the least amount of overlap. So we spot down all of our nanoparticles on paper, we quantify the RGB values shown here, and we calculate the overlap between each other. And so we can see, or non-overlap essentially. So we can see based on this heat map, the least amount of overlap is between Nanostar 450, which is this sample, and the red nanoparticles. Okay, so this is shown here. You can see that they're very distinct in color. Red and blue are very far apart in the color wheel, so this makes sense. And we take each of these and we conjugate them to antibodies of interest. So then we run the same heat map again with the blue nanostars, and we see again, we get a different pattern. And again, the most important thing is that some bind to yellow fever. Okay, some pairs bind to yellow fever and some do not. Okay, from this we do titration curves. This allows us to determine limits of detection, as well as look at binding patterns. So what we do is we incubate increasing amounts of NS1 for the 3 viruses, so dengue, Zika, and yellow fever. And we see this intensity increase with increasing antigen concentration, okay? And more importantly, the pattern that results differs depending on the antigen. So where we have dengue that's immobilized, and if we run it with red nanoparticles, we get high intensities. If we run it with the blue nanostars conjugated to Zika antibodies, we get lower intensities. So we can see already that we can differentiate on the between the antigens based on the antibody pair. Okay, so we're ready to put this all together. So what we do is we make a multiplex test. We mix our 2 antibody nanoparticle conjugates together. We have a red nanoparticle conjugated to polydengue. We have a blue nanostar conjugated to polyzika, and then we immobilize polyzika at this position and polydengue at this position. Okay, when we run it with dengue, we get a pattern that looks like this. So 2 red spots result, okay? And then this is the control spot in the back. If we run it with Zika, we get 2 spots as well, but they're different colors, okay? A purplish blue spot here and a paler one here. And then if we run it with yellow fever, we get a very, very pale spot here, but most of the signal is in this back one here, and they're both red. So you can see already just by eye that it's easy to distinguish between these antigens based on what appears in the test line. So you can just detect this by color. Okay, so to prove that this is really quantitative, what we do is we do principal component analysis to show that these are really separate. So we have 6 components, so RGB of this test line and RGB of this test line. And what we do is we do PCA, and we're looking at 2 components, and you can see already that Zika, dengue, and yellow fever all cluster in different areas. Okay, so these are distinguishable. Okay, then what we do is we train our system, using machine learning, and we feed it a true class, and we see what it predicts. This is plotted in a confusion matrix shown here. And so basically all of values are diagonal. So if you feed it dengue, it correctly predicts all 3 of them as dengue. If we feed it 3 samples of Zika, it correctly predicts them as Zika, and so forth. Okay, so this shows you that you can use basically dengue and Zika antibodies to be able to pick up yellow fever while still being able to distinguish it between dengue and Zika. Okay, we do this again for mixtures. This time we mixed yellow fever and dengue antigens, okay, in different ratios. You can see it's harder to tell these apart, okay, but again, with principal component analysis, we can see that we can distinguish between yellow fever, mixtures, and dengue. Okay, there are some errors in this, so it does mistake a dengue and yellow fever mixture for dengue and vice versa. So our overall accuracy is a little bit less, but it's still 87%. Okay, then what we try to do is try to distinguish dengue serotypes. There's 4 serotypes of dengue. They're all really closely related. We wanted to know if we could push this system to be able to distinguish between them. And so what we do is we run dengue 1, 2, 3, and 4. You can see that they all look fairly similar by eye. Okay, so we try to do principal component analysis, and we can see that based on the RGB values, it doesn't do very well. It actually mixes up a whole bunch. So it mistakes dengue 3 for dengue 1 and dengue 1 for dengue 3. Okay, so the RGB separation doesn't work very well. However, what we can do is take a trick from biology where they use pseudocolors. So this is something that's very common in biology. So biology uses stains that are not pure red and not pure blue. And so likewise, our nanoparticles are not pure red and not pure blue. And so biologists use basically a deconvolution called pseudocolors, where they separate the signal not into RGB values, but into pseudocolors that are indicative of each pure stain. So when you do this, we can decompose our colors into what is a color for a pure blue nanostar and the color pseudocolor for the pure red nanoparticle, as well as the background. And we use these as our components in our principal component analysis. If we do this, then our separation is much better. And you can see that there's only one mistake, or mistake's Dengue 1 for Dengue 3. So an overall accuracy of 92%. Okay, finally, we put this all together and we try to hack a test. So can we take a commercial dengue test that is out there, which is engineered to only detect dengue, and can we add more reagents to it, basically Zika and dengue antibodies, to have it pick up yellow fever? And so if we run our commercial test with yellow fever and Zika and dengue, you can see that it only picks up dengue, but not yellow fever and Zika. Okay, and so what we do then is we add additional reagents to it. So we spot down another dengue antibody on the test. Okay, we physically just immobilize this down. Okay, we add also to the running of the test our nanoparticle dengue conjugate, that's red, and then our nanoparticle, our blue nanostar poly-Zika antibody, and then we run it with the different antigens. And you can see that if you run it with dengue, you get a pattern that looks like this. This time, this is our hand-spotted area. Okay, if we run it with Zika, we get something that looks like this. So just the spot shows up. And then if we run it with yellow fever, we get a pale spot, but no line. Okay, and if we do the principal component analysis, now 3 components from the spot and 3 components from the line, we can see that we can separate these by antigen. Okay, and if we look at, if we use machine learning, we can see that we get 100% accuracy when we train the system. Okay, so this means you can train, you can hack a commercial off-the-shelf dengue test to be able to pick up yellow fever simply by adding more Zika and dengue antibodies. Okay, so you don't need yellow fever antibodies. Okay, and I do want to mention also that in this test, we don't know what these antibodies are. We just know that they bind to dengue, okay, and not yellow fever and Zika. So, okay, so with that, hopefully I've shown you that these paper-based immunoassays are very powerful. They have many advantages in that they're cheap and portable, and you can actually use the cross-reactivity of some of these antibodies to your advantage. And specifically, you can use them to repurpose antibodies to be able to configure a new test for an emerging pathogen. So we hope this will be an important tool for new outbreaks where there are no antibodies available. Okay, and the key to this is a little bit of machine learning as well as multicolor nanoparticles, and hopefully it can be used to be employed on emerging outbreaks. Okay, so finally, I'd like to thank my research group for this work, all really hardworking and talented people that I'm really grateful to be around. Results from this that I've shown you are from alumni, Cristina Rodriguez Quijada. This was in collaboration with Jose Gomez Marquez at MIT. I'd like to thank also my current group members, Dr. Delian Hristoff, who's a postdoc, Ham Rijal, who's a chemistry graduate student, and then Godwin Ujialale, who's an undergrad, as well as many of my past members, alumni as well. This work was funded by the NIH and also the UMass President's Office of Technology and Commercialization and Ventures, as well as UMass Boston. Finally, I'd like to thank you for your attention.